Thank you. All right, yeah, hi everyone online. To this end, we developed CM-impute, short for cross-species methylation imputation. CM-impute specifically seeks to impute a sample representing a species' mean methylation values within a particular tissue type, which I will henceforth refer to as species tissue combination mean samples, or combination mean samples for short. This type of mean methylation sample can be used to study any characteristics dependent on the variation in methylation levels between species and tissue types, and have been proven useful in studying characteristics such as maximum lifespan and gestation period. CM-impute is trained on different categories of observed data to learn the methylation patterns between different species and tissues. This toy example represents the data that could be used to impute these missing horse tissues shown in light green. The first category is same species, different tissue information, which allows CM-impute to learn patterns between different observed horse tissues. The next category is same tissue, different species, allowing CM-impute to learn the differences in methylation levels across species in the target tissue. The final category is overlapping species information, which allows cross-species methylation patterns to be learned between the overlapping tissue types in the target and non-target species. Altogether, this information allows the model to predict what the mean methylation levels are in the missing species tissue combinations. Each individual sample of the available methylation data, with the number of individual samples in each species tissue combination represented as the numbers in the grid, is inputted into a generative neural network framework called the Conditional Variational Autoencoder, or CVAE. This self-supervised model encodes the input samples into a latent space and then reconstructs the samples as accurately as possible. It is trained on two tasks: accurately reconstructing the samples, which is measured via binary cross-entropy, and regularizing the latent space to be as close to a standard normal distribution as possible, measured via KL divergence. The regularization allows the CVAE to generate new samples with characteristics not present during training. The key component of the CVAE is conditioning the input and latent space on a label, which provides additional information about each sample during training and allows for control over the types of samples being generated by the decoder. We specifically condition the CVAE on the species and tissue labels, which allows the model to learn weights for methylation patterns within and between different combinations, and thus be able to specify an unseen species tissue combination to be imputed. We chose to use this standard CVAE for this imputation task, as it provides a flexible framework that is able to generate new samples based on an arbitrary number of conditions. It also has the advantages of sharing information across probes and learning nonlinear relationships across various species and tissues simultaneously using a singular model. We select the hyperparameters via a grid search on a validation dataset. Once trained, the labels of the target species and tissue combinations can be inputted into the decoder, which, with the learned species and tissue relationships, imputes a combination mean sample representing the mean methylation level of each missing species tissue combination. To test CM-impute, we performed a 5-fold cross-validation analysis. For each fold, was randomly selected with the criteria that when a species tissue combination was held out, there was still same species, different tissue, and same tissue, different species data available during training. Here I show an array of all the held out observed samples. The samples and probes are both in hierarchical clustering optimal leaf ordering, which shows strong clustering based on phylogenetic order. We additionally visualize the observed data with the species signal removed, which reveals differentially methylated regions that are strongly associated with tissue type. When visualizing the corresponding combination mean samples imputed using CM-impute, we see similar methylation patterns to the observed data, with clustering across species and phylogenetic order, similar clustering of lower and higher methylated regions, and these differentially methylated tissue regions. Additionally, CM-impute achieves high imputation performance with an average correlation of 0.92 between observed and imputed samples across all probes. As a combination mean sample represents the mean methylation across individuals of the same species and tissue type, We show the natural variability among individuals within a combination as a reference for this performance. We additionally compare CM-impute to 4 baselines. The species baseline predicts the average of all training samples available in the target species. Logistic regression predicts the methylation level based on the species and tissue label. The tissue baseline prediction is the average of all training samples available in the target tissue. And the global baseline prediction is the average of all available training samples. CM-impute outperforms all these baselines. We further investigated CM-impute sample-wise performance across different phylogenetic orders. Overall, CM-impute yielded high sample-wise Pearson correlations across all orders, with the mean correlation within an order generally remaining between 0.877 and 0.935. The only outlier was the order Montremata, which only encompassed 2 out of the 907 combination mean samples considered in the cross-validation analysis. This demonstrates that CM-impute is able to accurately impute combination mean samples across the range of mammalian species. We next investigated CM-impute's probe-wise performance across all samples. We specifically focus on a subset of probes that are mappable in the majority of species. As a reminder, probes are considered mappable in a species if the probe sequence maps to a unique genomic location in that particular species, or there is a tolerated mutation. We define the subset of highest coverage probes as the 11,749 probes that are mappable in a significant proportion of the species with annotation files available. We specifically focus on this subset of highest coverage probes for the probe-wise samples, and this subset are less likely to be driven by differences in mappability across species and more likely to reflect within species methylation signals. On this subset, CM-impute achieved a mean probe-wise correlation of 0.623, significantly outperforming the baselines. We additionally evaluate how the observed variability across probes impacts performance. We consider here how much a probe varies across different tissue types. We use mean squared error for this evaluation, as Pearson correlation is less informative for lower variance probes. While CM-impute's MSC increases as probe variance increases, it is generally robust to both lower and higher variance probes. Specifically looking at the species baseline, which is the highest performing baseline, we see low MSC on very low varying probes. This was expected because if a probe does not vary from tissue to tissue within a species, then mean imputation within the species can be effective. However, the MSC increases quickly as probe variance increases. CM-impute additionally outperforms the other baselines. Overall, CM-impute has effective performance for probes across a range of lower and higher variance values. Once we validated the performance, we used CM-impute to impute 19,786 new combination mean samples for species and tissue combinations that have not been previously profiled, shown in magenta on the grid on the right. These imputed samples fill in the remaining 96.4% of potential combinations. While we do not have observed data to compare to, we see that the imputed samples cluster by phylogenetic order and show similar stratified patterns between the higher and lower methylated probes to what was seen during cross-validation. We further visualize the species and tissue signals present in the different datasets using the pairwise correlations between combination mean samples. The observed data exhibits a highly correlated block structure between samples of the same phylogenetic order. Within phylogenetic orders, there is an even higher correlation between samples of the same species. We do not see as obvious stratification between different tissue types in the observed data. While the full imputed dataset encompasses an entirely different set of species tissue combinations and cannot be directly compared to the observed data, it still exhibits a similar correlated block structure with the high correlation between samples of the same phylogenetic order and even higher correlation among samples of the same species. These patterns reflect what we would expect from the observed data. Next, we quantified these species and tissue signals of the full data using the area under ROC curve, analyzing the pairwise correlation's ability to predict whether two samples are from the same species or same tissue. We restricted this analysis to the combinations included in cross-validation to directly compare the observed and imputed results. CM-impute, shown in blue, yields a similar AUC value to the observed data, shown in red, for both the species and tissue groups. unlike the baselines shown in the remaining bars. Like the cross-validation analysis, we additionally visualize the imputed samples in probes with the species signal removed. As expected, these imputed samples additionally reveal differentially methylated regions, denoting that the imputation process is capturing more than just the species signal. To demonstrate that these differentially methylated regions are associated with tissue type, we visualize the pairwise correlations between combination mean samples with the species signal removed. We see that in the observed data with the species signal removed, samples of similar tissue types are highly correlated, with samples of the same tissue type being even more highly correlated. Once again, while we cannot directly compare the observed data to the full imputed data set, it still exhibits the correlation patterns we'd expect, particularly between similar tissue types and higher correlation within the same tissue types. This shows that the imputed data matches patterns we'd expect based on the observed data, and that the differentially methylated regions I showed on the last slide are strongly associated with tissue signals, and that we are capturing both species and tissue signals in this imputed data. As one example of a downstream analysis that can be performed with the imputed combination mean samples, we conducted a leave one species out linear regression analysis to predict the logarithm of the maximum lifespan based on methylation data similar to the Lee et al. Study shown here. We retrieved the maximum lifespan values from the ANH database. When using exclusively observed data, the predicted log maximum lifespan values yield a 0.81 correlation with reported maximum lifespan values. We then repeated the analysis using exclusively imputed data covering the same set of species and show that similar predictive performance was achieved. Additionally, the final predicted lifespan values using observed data and using imputed data exhibit strong correlation with one another, demonstrating that not only are they achieving similar performance, but the actual lifespan values being predicted are similar. This shows that the imputed data is a valid substitute for the observed data. In conclusion, the development of the mammalian methylation array greatly expanded the amount of available cross-species methylation data, but left gaps in species and tissue availability. To address these gaps, we developed CMImpute, which uses a CVAE to impute combination mean samples of missing species and tissue combinations. CMImpute yields high imputation accuracy and maintains species and tissue characteristics seen in observed data. Finally, imputed combination mean samples can be used for downstream analyses, such as predicting maximum lifespan. I'd like to thank everyone in Ernst Lab and Steve Horvath, Cesar Lee, and the Mammalian Methylation Consortium for collecting all of the data and sharing it with us, and all of our funding sources. You can find this work up on bioRxiv at the preprint, and I'd be happy to take any questions.
Hi, very cool method, and I like the application to the maximum lifespan problem. Are there other applications you're thinking of that one could use the output of this imputation for? Like what else could someone use that for?
Mm-hmm. So I think that generally the aging applications are the ones that are most studied currently with these mean methylation samples with the aging and gestation period. I think what is cool about like these imputed samples is they're also tissue specific. So I think differences in like cell type specificity across mammals and things like that could also be an interesting study, particularly if these tissues are similar or different across mammals, and if those correspond with any other changes across the species, would kind of highlight the tissue specificity of them and be interesting to look at.
Fascinating. So methylation is dynamic in some tissues. I'm curious if the errors in your imputation correlate with CPGs that are more dynamically methylated. Or is there anything? Are there any signals of which ones change more frequently than others?
Do you mean dynamically within different individuals or across? Yeah, yes, yeah. So I think that that's definitely probable, since we're looking like not at individual specific samples. I don't have the exact like data on that, but I think that in general, yes, like when we're performing slightly worse than others tends to be on more highly variable probes. And while we're still outperforming the baselines, this is something to note, and that I do believe that it would be on more dynamic probes at times.
Thanks. Great talk. So if I got it right, species is the dominating factor over tissues.
Yes.
And that's unlike RNA and expression or splicing where
Mhm.
tissues dominate. Is it all explained by lifespan, or how come species is so important for DNA methylation?
It's an interesting question. I don't know if I have a great answer because that's just in the observed data, and so it wasn't like my primary focus. I think because DNA methylation does have a strong impact on aging, that there might be some sort of difference in that either it causing the aging or like vice versa. That might be causing some of the differences across the different species. But I'll be honest, I'm not 100% sure. That's a really good question. Yeah.